Good afternoon, everyone. Welcome to the Tufts Research and Data Symposium for Food and Nutrition 2022. Really excited to be kicking off with one of the earlier sessions for our 3-day symposium. So my name is Emily, and this is the Precision Nutrition and Personalized Health panel and panel discussion. And we are delighted and looking forward to what's to come. But just a few housekeeping things. This session will be recorded. So if you would prefer to keep yourself muted and to keep your camera off, please do so. We will also have our panelists present, and thereafter at the end, we will do a panel discussion. And that will be the time in which you can submit your questions either via the chat or in person. And we'll just go with how many questions we have, depending on how much time we have thereafter. So to start the session, we have Dr. José Ordovás. Dr. José is a senior scientist and lead of the nutrition and genomics team at the HNRCA. And we're very lucky to have him here to discuss today because he works in areas of nutrigenomics related to the regulation of gene expression in microRNAs and DNA methylation, and the relationship between genes, chronobiology, and obesity. So Dr. Ordovás is considered one of the founders in nutrigenomics. So he is going to kick us off today with this discussion.
Thank you, Emily. Today's session is going to be dedicated, as Emily mentioned, to precision nutrition. And what I'm going to do is not to give you the basis or the foundation of precision nutrition or nutrigenomics, as we used to call it in the old times, but to move in an area that is that also it has been mentioned, which is the chronobiology of nutrition, and how, as the title indicates, precise timing is also a critical piece of precision nutrition. And why precision nutrition? And this is a theme that will be repeated over and over, is because that approach to nutrition, the one size fits all, well, it's not working as expected, right? And we can see that, for example, in different parts of the world, people are eating at different times. And in this case, I focus on dinner time. Then you have countries like the Scandinavian countries, where you have dinner from 4 to 5, whereas on the other side of the spectrum, you have countries like Spain that they go from 9 to 11, and everything in between. So you see that in terms of also defining the cultural aspects of nutrition, one size does not fit all. And this is important because of our rhythms, our biological rhythms that are represented in this slide. So at every moment of our days and nights, we are different. Not only different with other people, different among ourselves, right? And then we perform for some things, it's better to do it in the morning, some things are better to do it in the afternoon, and something like this, sleeping is better to do it in the night. So a topic that is also of great interest to me is precisely the sleep and how we are disrupting this healthiest sleep that we should all be taking, either by our lifestyle or because of shift work and so on. So anyway, in terms of what I was saying earlier about the dinner time, you can see that in different parts of the world, some people eat at very different times that gets us in different points of our chronobiology. And that may be of a great impact in terms of public health. What determines our chronobiology? Our chronobiology is complex and is dictated by what we call the circadian gene network. which we see in the left part of the slide. And the discovery of this network is relatively recent. And first, we were focused on the central clock, which is in the suprachiasmatic nucleus in the brain. But then with time, we discovered that every one of our cells, organs, they have also their individual clocks that in the best case scenario, they will be synchronized with the central clock in the brain. And we have two scenarios. One, that the synchronization is good. Then we have a circadian regulation of sleep, metabolism, the microbiota, the cardiovascular system, aging, in fact, and cancer. But if there is chronodisruption, the consequences are serious, because then we get into, for example, in terms of sleep, poor sleep, and the consequences will be also depression and so on. In terms of metabolism, obesity, type 2 diabetes, in terms of the cardiovascular system, atherosclerosis, cardiovascular risk, and so on, so on, so on. So time is very important in our biology. When we do things is really important. So that takes us to the fact that we are not only what we eat, but also when we eat. And then I am going to give you some examples of, for example, the chronotypes. And there are people that are larks, there are people that are owls. And what we have, this is a very recent publication from the CardioPrep study, which is similar to PREDIMED, but in this case with people that had previous cardiovascular disease. So it's a secondary prevention study with diet. And then what we did, it was to divide people among chronotypes, evening or morning chronotypes. Let's look at the baseline of the study before they went into the different diets being a low-fat diet or a Mediterranean diet, just like in the case of PREDIMED. So what we can see is that at baseline, evening type people had higher triglycerides, lower HDL, higher CRP, meaning inflammation, and also higher homocysteine. So the cardiometabolic risk factors were elevated in these people at baseline. They were at higher risk. They were put for several years into either the low-fat diet or the Mediterranean diet. And you can see that there were some effects, but even after being in a prudent diet or either one of the prudent diets that were used, the chronotype was overpowering the effect of the diet. And always the evening type people, they had a worse profile than those that were morning type. So this reinforces the importance of our lifestyle and our chronotypes in terms of cardiometabolic health and also other systems like neurological and so on, cancer, that were not examined in this study. So another aspect of more related to the timing of food comes from this other study in which we look at the response in terms of body weight loss resulting from a Mediterranean intervention, different from the CardioPrep. And what we could distinguish is two different patterns, one in which the weight loss were less successful, and in other individuals it was more successful. They were consuming the same, but still the results were different. What was the difference between these subgroups? Well, the difference between the subgroups was the time in which they were eating their main meal. And that in the case of the, let's call it poor responders, they were eating later, and the good responders were eating earlier. So again, this reinforces almost that the advantages of that morning chronotype. And that was reflected also in terms of diabetes risk with HOMA. And also, we could observe that there was a genetic component. It was not only the social upbringing and where you are, but also a genetic component that was driving people in one chronotype or another. So what we can say is that from the, I would say, public health perspective, let's recommend people just to move their habits earlier, to an earlier time, to move that chronotype from evening to morning. Well, probably that could be a good idea, but is that going to be something that benefits everybody? And that's where precision chrononutrition comes into place, because in the same study that I showed you before in terms of weight loss, depending on the meal time, then we added the layer of genetics, and you can see how the results are really neat, because then looking at this polymorphism in the perilipin-1 gene, that is a gene that is expressed primarily in the adipose tissue, then we can see that if you are a T carrier, you have the T allele of that polymorphism, it doesn't matter if the time is not so important in terms of weight regulation. But for those that have the genotype AA, it's really, really important, as you can see by how you spread the difference in weight loss between those that ate late from those that ate earlier. So whereas you can say that it will be a good public health recommendation, I think with the knowledge that we have, we can go deeper into the concept of precision nutrition. And yes, in the future, we'll be able to say to people what they should be eating. But also we can play with the chrononutrition, with the precision chrononutrition, in order to give a recommendation also about when people should be eating or how they should be distributing their meal during the day. Here we have another example, and I'm going to finish very soon because of time moves very fast. And here we have another gene, which is the melatonin receptor. And here what the investigators did was for people to have their dinner late or dinner earlier. And what you can see is that the effect on the glucose levels, the postprandial glucose levels, time didn't matter if you were a CC, had the CC genotype of this polymorphism, and you can see it in the lower part of the right panel. In the upper panel, you can see that now, if you are a GG, that's different, because if you eat your dinner late, then your glucose spike is going to be much higher. And if you do that day or night after night, obviously that is going to put you on a higher risk of diabetes. Whereas for these people, they will benefit a great deal if they had the dinner earlier, as you can see by the gray line that is on the upper panel of the slide. So again, this is another example of how in the future we'll be able to give recommendations based not only in what we tell people what to eat, but also to identify people that will benefit in terms of modifying their when they are eating their meals. Obviously, this is a very dynamic process, and we will not be able to bring this to the population and to have success if we don't take advantage of integrating the classical nutrition with the new technologies that are going to be, I would say, essential in this type of precision nutrition, which is so dynamic and is capturing our status, nutritional status, but also all the other parameters of interest, real time and continuously through our days and nights. So that is, I would say, a development that we have to contribute to and follow very closely. And obviously, what we have done so far is just to, I would say, paving the way to what is happening as we speak in terms of this new program of nutrition for precision health, powered by all of us, which has been an NIH initiative in which TAFs is an intrinsic part of this study, also in which during the next 5 years we'll be able to bring all these technologies to a level that we were not able to do in the past because of the limitations, mostly in terms of population size that we had. Now, with $170 million, we can start thinking about bringing that to thousands and thousands of people rather than hundreds as we were doing in the past. So we have great hopes that 5 years from now, we will be in a very different position when it comes to precision nutrition than we are nowadays. And with this, for me, it's time to say goodbye or see you later after we hear the other wonderful speakers that are lining up. Thank you so much.
Thank you so much, Dr. Ordovás. That was super interesting, and I have a question, and I saw there was a question in the chat, but we will get to them at the end. Next up, we're going to have Dr. Sheldon Rowan. Sheldon is a scientist at the Nutrition and Vision Lab at the Jean Muir USDA Human Nutrition Research Center on Aging and Tufts University. And he researches the role of dietary carbohydrates in the development of age-related macular degeneration and its relationship to aging, metabolism, and the gut microbiome. So thank you, Sheldon.
Okay, thanks so much, Emily. I'm really excited to present a little bit about my data today and kind of talk to you about what we can learn from the mouse as a model to understand precision nutrition. I'm just going to give a very, very brief introduction into kind of like the kind of research that we do and how we ended up studying the microbiome. So our group is interested in understanding dietary relationships to age-related disease. In particular, we're interested in a disease called age-related macular degeneration, which you can see here compared to normal vision, kind of strips away your central vision. It's the leading cause of blindness in developed nations and a huge growing problem. And as a nutrition researcher, we're interested in relationships between dietary patterns and risk for disease. And what we noticed a long time ago from epidemiological studies was that individuals that consumed a high glycemic index diet were at increased risk for developing macular degeneration. But the mechanisms were completely unknown. So what I sought to do when I joined the group was to establish an animal model where we can try to potentially both recapitulate the findings in humans and get mechanistic insights. And we also wanted to do this in an aging context, which is extremely hard to do in human studies. So we developed a wild type mouse study where we age mice from 12 to 24 months, kind of corresponding from early middle age to older ages, where we feed them matched diets. They either consume a low glycemic diet here or a high glycemic diet. The only difference here being the starches. And then we have a dietary intervention where midway through the study in this crossover group, we switched the mice from a high to a low glycemic index diet. And what we saw, which was really exciting at the time, is that the diet that we fed the mice had a big impact on the risk for eye disease. So animals fed a low glycemic index diet maintained a healthy eye. You can see this both photographs in the back of the eye and through the retina. Animals fed a high glycemic diet developed eye disease that resembled age-related macular degeneration. And if we switched the diets midway through the study, we were actually able to rescue eye health throughout the study. So we kind of have established this firm relationship between diet and eye disease. Where does the gut microbiome fit into this? And so we decided to look at this in a kind of longitudinal sense. And the first thing that we found, and you can see here, these are beta diversity plots, where the closer plots are together, the more similar the gut microbiomes look. Keeping in mind that these are all genetically identical mice. So we're kind of like trying to understand what are the sources of variation. And diet is a huge source of variation in the gut microbiome. So animals fed a low glycemic diet have very different microbiomes than those fed a high glycemic index diet. And we could get some sense for diet as a biological variable by our group where we switched the diets. These are the ones going from the green to red. And you could see in a couple of months, some of the gut microbiota completely transformed to look like those that they're now consuming a low glycemic index diet, whereas others take a longer time to transition until eventually all of them resemble each other. So the diet certainly changes the gut microbiome. This is well established. But in the context of aging, we saw some interesting effects. And so I'll show you a couple of examples of this. The first is from that original study that I showed you. We were able to actually identify specific gut microbiota that didn't care about what diet they were fed, but they increased with aging. So in all of the diet groups, the bacteria identified on this principal component analyses increased with aging. And this was kind of consistent with data coming out from humans looking at what happens to the gut microbiome through the aging process. These are with, mostly static samples and comparing cohorts. And then Emily, in her practicum with me, did an interesting study where she looked at what happens to very young mice fed these same diets compared to old mice fed these diets. And what she found, this was quite striking, is that not only do the young and the old mice have quite different looking gut microbiomes, but the dietary effect was highly modulated by the age of the mice. So the young mice, you could see, have like a very large difference in the diet. And the older you get, the more attenuated these differences become. So there's certainly aging is an important factor in looking at what happens to the gut microbiome. We're also interested in understanding the role of genotype in risk for both macular degeneration and its relationship with gut microbiota. And just quickly, I want to tell you about another study that we did where we took mice that were deficient in the NRF2 transcription factor. This is a gene that's involved in controlling antioxidant repair pathways. We fed them these same diets and aged them. And the mice fed a high glycemic index diet, developed actually quite severe eye disease at a younger age than we see in the wild types. You can see in histological section, these developed quite advanced disease, whereas the mice fed the low glycemic diet maintained their retinal health, looking almost indistinguishable from wild type controls. So we wanted to know if any of these genotype diet interactions were affected at the level of the gut microbiome. Without a priori having any knowledge about what NRF2 itself may be doing to the gut microbiome. This is now work that a graduate student in the lab, Kelsey Smith, together with a master's student, Shuidi Wang, did, looking at the role of NRF2 now in the gut microbiome. And you can actually see that we get quite a significant effect of genotype on the gut microbiome. We still maintain diet relationships, but the role of the genotype is actually even more significant, with the largest divergence between the wild type mice fed a low glycemic index diet and the mice from NRF2 knockouts fed a high glycemic index diet. These are the ones that develop the most extreme eye disease. And so we've been going on to try to understand this on a more functional basis, looking at pathways that are affected by these changes in the gut microbiota. And what we see here is when we start looking at the functional units that are controlled by these gut microbiota, the divergence becomes even larger, whereas the wild type animals actually look relatively similar functionally. The NRF2 knockouts start to show huge divergence, and there's unique pathways that become activated in the absence of NRF2. So we're now trying to dissect these out using network analyses. We're looking at relationships both between metabolites, microbiota, and their various pathways. And I think this will be really important, illustrative in understanding all of these different roles for biological variation in gut microbiota and how those contribute to precision nutrition. Thanks.
Awesome, thanks, Sheldon. Yeah, I certainly learned so much whilst working on that with you as well. Okay, next up is Sylvia Benitez. Sylvia is a nutrition researcher here at Tufts, and she focuses on the area of precision nutrition and studying the interactions between factors that influence human behavior and health outcomes. But not only that, she's had experience developing a platform called Serenist, a multi-omics integration platform that optimizes prenatal nutrition, and has also co-founded Berciano Foods to market better products. So we're lucky to have you today, and thank you, Sylvia.
Thank you, Emily,
No
for your
worries.
introduction. It's a pleasure to be with all of you here today. So here today, I'm going to talk about precision nutrition across the lifespan And I'm very pleased that we've already heard a little bit about different stages in our lives and how different omics play a very important role in precision nutrition, trying to identify the role of genetics, microbiome, and other different omics influencing our health and reflecting our health status and dietary responses. So I'd like to start by defining precision nutrition as an approach that utilizes individual data to accurately predict how a person responds to different foods or diets, leading to more effective recommendations to help the individual achieve specific health goals. So in contrast to more traditional approaches, precision nutrition uses this data, individual data, which can comprise biological factors such as anthropometrics, genetics, gut microbiome, health status, including medical conditions, nutritional status, metabolic health, and also lifestyle factors such as our physical activity, sleep, medication use, smoking, and also diet. And diet includes not only what we eat, but also the timing of our meals and the context in which we eat, as José described earlier today. And this information is collected using different methods, from questionnaires to wearables to analytic tests, and then integrated. We're relying more and more on artificial intelligence for the integration part because of the amount, the depth, the width, and the variety of variables that we're collecting. It's big data. And then that results into personalized recommendations that are returned to the individual and used to promote a certain behavior change. And then the cycle repeats again, and we close the loop, measuring the changes, measuring the resulting diet, the changes in lifestyle, the changes in health outcomes, and everything starts again. So this approach is personalized because it uses this individual data. It's reliable. The recommendation should be more effective than the conventional one-size-fits-all approach. It's evidence-based. It needs to be supported by data and scientific evidence. It's complex because all of these different integrations are required for its development. It's very difficult to produce a precision nutrition approach only looking at genetics or only looking at the gut microbiome, because we've seen and we'll see later how these single variables often predict a very small percentage of the variance in those dietary responses. It's also integrative. It brings together all of those multiple predictors, systematic, because the data is collected, integrated, and presented systematically. And very importantly, it is evolving because those dietary recommendations evolve over time as the person changes or additional evidence becomes available. And since we've talked about a little bit about the different stages of life, I wanted to highlight today how we change as we age and how precision nutrition may play a role in each of these different stages. So in the last few years, collaborating with innovators at MIT and Harvard to develop a company called Ethos Genomics and our Serenis platform, we wanted to look at a problem that we started seeing among our friends when they were trying to start a family, planning to conceive. And many of them faced challenges with their fertility. And it was very shocking for me to learn that 50% of the couples under 35 that undergo IVF don't go home with a healthy baby. And some of those challenges for fertility and for conception are due to diet and lifestyle. But still, in the fertility clinics, diet is less than an afterthought. So there are many different recommendations in terms of stress and lifestyle and pharmacotherapy. But diet is not yet sufficiently utilized and studied to overcome some of these challenges that our generation faces. And since we also know that diet starts before conception with the health of our parents, and we know how important the whole prenatal environment and prenatal development is for our long-term health, I thought that it was interesting to start thinking of how precision nutrition could be part of this approach. And the method that we ended up developing was quite simple and practical due to the limitations of costs right now. But we used a genetic panel that looked at both the carrier status of the mom verified with the father if genetic conditions that were threatening were found. And we also looked at genetic variants that represented a susceptibility factor for preterm delivery, for preeclampsia, for gestational diabetes, different things that if we know in advance, we can use to develop more personalized dietary recommendations for pregnancy and lactation. So we worked with a team of dietitians, and we also had some blood tests performed, mostly looking at nutrient status to correct deficiencies and insufficiencies. It wasn't a metabolomic panel, but we will see that as precision nutrition evolves, those tools and approaches will become more accessible and also more applied in every single life stage. Right now, we focus mostly on one-size-fits-all approaches. So we can look, for instance, at My Plate, which has specialized recommendations, more stratified plans based on the dietary guidelines for Americans for different life stages, but they are still very general. So they are focusing on very well-known facts regarding the process of food introduction, breastfeeding or formula feeding, developing food acceptance and in healthy eating habits in infants, in children. And then, you know, as we're highlighting here, our metabolism change, our priorities change, our diets change. And it can really be used to correct some of those health progression and disease progression trajectories if we have the tools to identify those early changes, early processes and early markers of disease, and more personalized interventions that can help us promote health for every mom, every baby, every child, and every adult. We've talked briefly about the role of genetics, and I think it's always good to review why we care about genetics, because sometimes there is this unfair perception that because it's in our DNA, it cannot be changed unless you do some extreme biohacking or genetic editing. But our genetics contain a lot of information about us and about our health risks. They also contain information about how we taste, our dietary preferences, our behavioral traits such as hunger, satiety, emotional eating, things that when our dietitian is aware of those predispositions, they can be addressed and they can be used to develop a more personalized plan that will not only contain the recommendations that are better for us, for our metabolism, for our health, but also incorporate some of those behavioral aspects that will make the plan, the dietary plan and recommendations more aligned with our lifestyle and easier for us to promote that compliance. And then finally, we have gene-nutrient interactions. So those are mostly what we refer to when we talk about the drivers of different dietary responses. And it simply means that the expected response to a nutrient intake or to a diet depends on whether or not you have a specific set of genes. And this, again, knowing about this can help us understand what would be the expected outcome if we make a specific change in our lifestyle, in our diet. Then we've also talked about the gut microbiome, and I think it's important to highlight that this is a very, very young field, and there's been amazing advances from finding that the microbiome has a causal link to obesity. And for instance, a fecal transplant can directly cause weight loss. But also we have very recent research that has identified the mechanisms through which a specific type of bacteria in our gut, the Bifidobacterium infantis, can drive systemic inflammation and immune system imprinting early in life. So Bifidobacterium infantis is a type of bacteria, is a type of Bifidobacterium that can digest the human milk oligosaccharides. And it's fascinating to learn that most women in developed countries have lost their Bifidobacterium infantis. And therefore, unless they take probiotics, they don't really pass it to the baby, and the baby cannot make use of all of those oligosaccharides that are in the milk and present a very high percentage of the calories from milk. So it's interesting, and it's very well aligned with what we know regarding how the mode of delivery, like C-section versus vaginal birth, or the breastfeeding or formula feeding can impact the infant microbiome and health later in life. So we can have a huge discussion regarding how our microbiome evolves throughout our life. But to summarize, we know that the variety of species in our gut increases as we reach our maturity, and then declines with age in general. But that's very personal as well. And there are a lot of gaps in the research, although we know that the microbiome is relevant for all of us, even if we don't have a specific metabolic disease at the moment. Research such as the one highlighted in the CV et al. Paper that later resulted in the company Day 2 can help us predict dietary responses, glycemic dietary responses to different foods. And before we looked at glycemic indexes, glycemic load, and everything was kind of, like general standardized. But if we look at the individual, we may see that something that is expected to have a higher glycemic response, such as eating a cookie versus a banana, may have a lower response for many individuals, depending on the gut microbiome. That said, we're still not quite there yet. And we're currently moving from studying just what's in our gut, the taxonomy and abundance, our gut microbiome composition, to what is it doing, the functional profiling. And a lot of us remember the earliest studies that were looking at, like Bacteroidetes and Firmicutes, and trying to understand what is expected in terms of weight loss based on those very large groups of bacteria. And now even looking at more specific types of bacteria in our gut, we're still lacking key information, because the bacteria, just like nutrients in our diets, don't operate in isolation. There is an ecosystem in our gut, and bacteria interact with each other to produce short chain fatty acids and different signals that end up shaping our health. And we also know that there is no single profile for a healthy microbiome. In the microbiome of completely healthy people, based on all the markers that we use right now, may look very different based on where they're from, their diets, their lifestyles. So it's still an area under development. And when we are looking at, yes,
So I'm so sorry. We were going to have to cut you a bit
Oh.
short because we'd still like
Perfect.
Dr. Brisboisis to talk. I wish we could continue past 2 p.m., but we won't be able to. So I'm going to have to cut you short there, and I'm so sorry about that, everyone. But please reach out to Sylvia if you do have more questions for her, because we are running a bit short on time. So next up, sorry about that. Next up, we have Dr. Tristin Brisboisis, who is the director of the Advanced Personalization Ideation Center for the PepsiCo Life Sciences. Yeah, thank you so much for being our last speaker today, and hopefully we get through your slides. Thank you so much.
Yeah, no problem at all. Thank you for the invitation on the panel. And I guess it's my job to provide a bit of an industry perspective to the challenges and opportunities in personalized nutrition. So we're defining personalized nutrition quite simply as answering this question: should I eat or drink this or that? And based on the fact that more consumers are asking this question, we've seen kind of two trends emerging, particularly the past year. One is with retailers. And so you see a lot of retailers across the globe really incorporating nutrient profiling systems to offer personalized recommendations for healthier options. And these can either be through their digital space and e-commerce, and they're using different behavior change techniques to really encourage healthier behaviors. And again, you see this across the globe. So Kroger in the US, you have an example from the Netherlands as well as Latin America. The other trend you see is that there's been a huge uptick in wearables. And so what's interesting about this is that there seems to be sort of a race for the next health feature. And there's definitely a shift to more of this illness prevention, or getting away from being just in reactive and tracking mode and actually getting into more prevention. So you see this with the new Apple Watch, right, for fall risk. And so, we move past this just tracking of steps, even energy expenditure, that's almost table stakes. And so it's quite interesting to see how that has moved into more of an interpretation. Rumors have it that there might be a blood glucose, continuous blood glucose monitoring as the next feature, which offers an interesting opportunity, but also some potential challenges. And it'd be great to get this panel back together at some point to be able to discuss those, because I know both Jose and Sylvia share the potential concern with that, and also the opportunity as well. Now, in terms of what is happening in the marketplace when it comes to diagnostic tools and different tests that are available to consumers right now in the personalized nutrition space, these are quite expensive, and they're also onerous, often onerous for the consumer. And so that is problematic for two reasons. One is that it's really tough to scale because of the cost associated with it. And then you're also having the consumer weigh the potential effort. Is it worth the insights that they will be getting in return from completing this diagnostic test? And so really a huge unlock, I think, within personalized nutrition would be to develop wearables or diagnostic tools that are really accessible to all in a non-invasive way. And that will really get personalized nutrition to that next level that we need for it to reach the masses. How we've tackled the personalized nutrition at PepsiCo, we've started with the athlete. And so we are looking at or have developed a sweat patch, and that is measuring whole body sweat, but just from the forearm. And so it's measuring your fluid losses as well as sodium losses. And this wasn't a project that happened overnight. So the team went through 5 years, 11 clinical trials to validate this patch, ensuring that it's measuring what it's supposed to be measuring, and also that you can get whole body sweat just from the one spot on the forearm. And so it's available now to, you can go pick it up anywhere in the US in any sort of sporting goods shops. So that's fantastic example of how science has translated into a commercial success. And there was a lot of iterative processes that had to happen with this sweat patch. So it wasn't just about the clinical trials to validate the wearable, but it was also, you had to, one of the original prototypes, the flow of fluid didn't quite go fast enough. And so then, to tighten up those tubes so that they would have a faster flow that would also go further down the patch. And that just provided a better consumer experience. Because at the end of the day, consumers have to be engaged in this space in order for any behavior changes to actually take place. Sorry. So that, for PepsiCo, that's sort of where we've started our journey again with the athlete, and that GX platform will continue to evolve. But they're also presented the question of what about the general population and what are the opportunities for the rest of the portfolio? And so that's really what my team is set up to do. We recently launched what's called the Advanced Personalization Ideation Center, or the API Center. And our mission is to deliver actionable and eventually profitable personalized nutrition solutions to promote the health and wellness of our consumers, and to do this in a fun and engaging way. And so we operate very similar to a startup or a learning lab, meaning that we rely very heavily on external partnerships, so both in the academic standpoint, as well as other industry members. And the reason we do that is so that we have a very agile approach. And so we don't have to invest necessarily completely on developing one specific capability, but we're able to test and learn different technologies, different solutions, see what works, and then be able to invest in scale. And so it's a very agile way of getting products and services into the hands of consumers quicker, but not foregoing, the scientific rigor that is associated with it. And so if we look at what has happened in the space of science over the past recent years, science of the omics, so genomics, proteomics, the microbiome, all of those areas have just increased in terms of research, and they all produce very large data sets. And so the advancement of analytics has helped, bring those sciences to a new level, because we're able to use machine learning or AI to really be able to garner the right insights out of those particular scientific fields. And what has also happened in parallel is that there's more of a technology adaptation or adoption. And so people are using wearables more and more. And so what you have now is a bit of a critical mass of data that's available in the real world. And so that's really interesting and exciting, because you could almost have a bit of a hybrid model where you have, access to this real world data, as well as being able to validate in a clinical setting. And that's really the approach that the API Center will be taking going forward. So just to finish, I might have time for one or two questions, really to talk about the future direction then, or as I see it for personalized nutrition. There needs to be a balance. So there needs to be the ability to implement standards that are able to maintain consumer trust as well as a scientific rigor, but not at the expense of innovation. So innovation in personalized nutrition is operating at an unprecedented pace. On the other end, traditional research is quite slow, regulations are even slower. And so, potential move forward is really to think outside of these traditional methods. And again, to highlight just the importance of these different consortia that operate in the pre-competitive space, as they're looking at industry-wide standards or setting guiding principles that will allow that kind of nimble and agile approach, because a lot of startups, or a lot of the place in personalized nutrition, you have to learn as you go and build out those models. And so how can you do that by still maintaining the integrity of the science, as well as ensuring consumer trust is maintained? So just a couple of the different consortia are listed across the bottom. Obviously Tufts has one here as well. And so with that, I'll turn it back over to Emily if there's any time for questions.
Awesome. We unfortunately don't have any time for questions. I really wish we did, but I just want to say thank you so much to everyone who attended. And thank you so much to our amazing panelists. And I see there's been some emails put out in the chat if you do want to reach out to them. Again, apologies we didn't get to questions, but this was really great. And I just want to thank everyone for being here. Thank you.